Hello students, welcome to the session. Students, in this session we are going to discuss about the assignment key dated on 10th July 2020. Children, in the previous session we have learnt the assignment key for the microscope. In this session we are going to discuss some of the questions given in the assignment of 10th dated, and let us see the questions whatever given in the assignment. First question I am reading. What is the purpose of staining? What is the purpose of staining? What do you mean by staining? Before going to learn purpose of staining, you have to know what is staining. Addition of colors to a slide or to a specimen is called as a staining. You know that purple sulfur bacteria or lactobacillus bacteria. If you want to see the lactobacillus bacteria in front of microscope, we need to add crystal violet color. What is that crystal violet color? When you add crystal violet color to the lactobacillus bacteria, the plasma membrane or cell membrane which is there around the bacteria absorbs that crystal violet color or a particular color and reflects or visible in front of the microscope. So, the purpose of staining is it helps to observe microscopic organisms or microscopic living creatures that are present in and around our surroundings. Now, let us see the purpose of staining. So, how can you give the answer for purpose of staining? My answer is, one gunshot answer is to observe microscopic organisms in a proper manner. The staining process plays an important role. Very simple answer. So, to identify or observe smaller creatures, smaller organisms. What are smaller organisms? The microscopic organisms which are not visible to our naked eye. If you want to look at them or if you want to see them, we need to go for the staining process. Purple sulfur bacteria, by adding purple color, they are visible under microscope. So anyway, staining process helps for identifying microscopic organisms that are there in our surroundings. Students, I am moving on to next question. What precautions are needed when preparing a microscopic slide? Already, while explaining about different types of experiments that are given in the cell, the basic unit of life, I had explained the preparation of microscopic slide. So, the precautions are, second question, what are the precautions you have to take while preparing a slide? We should not add excess of dye. What do you mean by excess of dye? What do you mean by dye? The dye is nothing but color. We should not add much color. We should not add much glycerin solution. We should not add much water or water droplets to a specimen that we are taking on the slide. Suppose take a slide. The generally microscopic slide looks like this. Suppose I want to observe the amoeba or a paramecium present in a sample of water. I have taken a drop of sample of water on a microscopic slide. After taking the drop of water, sample drop of water, then what we have to do? We need to add the color or dye. After adding dye, what we have to do? We need to add one drop of glycerin. Finally, we have to place the cover slip. So, first precaution what you have to take? We should not add excess color, and then we should not add much water droplets. Then third step is, without air bubbles, we have to place the cover slip on glass slide. If there are air droplets below this cover slip, what will happen? The specimen or a slide could not be visible under microscope easily. Hence, So these three precautions are very important. Next, students, go for the third question. How do you prepare a microscopic slide? Very simple question. How do you prepare a microscopic slide? Third question is, how do you prepare microscopic slide? What is your answer? What is your answer? How can you prepare the microscopic slide? So first step, we have to clean the microscopic slide properly. After that, take the sample on the glass slide. After that, add 1 drop of water. After that, add 1 drop of glycerin. After that, add 1 drop of dye or color to the specimen. Finally, with the help of needle, slowly we have to place the cover slip on the slide. Okay? So if you use the needle while placing cover slip on the specimen, what will happen? Air bubbles do not occur. That leads to clear visibility of specimen under microscope, isn't it? So these are the different steps that we have to take while preparing the slide. Suppose example, I want to give an example. Already we have learned many examples. For better understanding how can we prepare a microscopic slide, I would like to present an example. Suppose if you want to see the blue-green algae that are present in the pond water, how can you prepare the slide of blue-green algae? First, collect some sample of water from the stagnant water or from pond or from stream. After collecting the stagnant water, take a drop of sample, take a drop of water or sample on microscopic slide or glass slide. After taking a drop of water and slide, immediately what we have to do? Then we have to add a drop of glycerin. We have to add a drop of glycerin. After adding a drop of glycerin, finally we have to add a drop of any dye. We can take a phenolphthalein indicator, or we can take a methylene blue solution or Janus green base solution, or any dye or color we can add. After adding color, what we have to do? Finally, take a cover slip, place it on the specimen without air bubbles. Take a cover slip, place it on the specimen with the help of needle without air bubbles. So, in this way, a proper microscopic slide can be prepared. So, these are the events or steps that are commonly seen in the preparation of microscopic slide. So, with one more assignment key and assignment answers, in the coming classes we are going to learn. Thank you. Have a nice day.